Hi, can somebody confirm you can hear me?
We can hear you.
Thank you. Before we begin, are there any questions? All right, let's begin. Today is January 28th, and we are supposed to be finishing chapter 3. We actually finished it last time, and beginning chapter 4. Let me remind you, you do have a quiz this Wednesday. The quiz will be covering chapter 3, the end of chapter 2, and labs 3 and 4. Any question about that? All right, last time we had talked about special stains. I did tell you you do not need to know special stains for the quiz and the exams, but you do need to know it for your unknown project because you may need to do a special stain for your unknown project. Any question about special stains? All right, let's get out of here and begin chapter 4. Chapter 4, the functional anatomy of prokaryote and eukaryotic cells. Now, let me warn you, normally I do not cover the anatomy of eukaryotic cells because you should have had that in your preliminary biology class, which I believe would be for most of you, biology 160. I will cover the functional anatomy of prokaryotic cells, but you should already know the functional anatomy of eukaryotic cells. In the rare case that I have time, I will begin the discussion of the anatomy of eukaryotic cells. I will only talk about one thing. Well, I will for sure talk about one thing in the anatomy of eukaryotic cells, and that is the endosymbiotic theory, the theory to explain how mitochondria and chloroplasts came about in a eukaryotic cell. All right, so this slide is showing you the major goals and the rough outline of chapter 4. You need to know the terms. What's a coccus, a bacillus, a spirilla? You need to know what strepto means, like streptococcus, staphylococcus, diplococcus, a tetrad of cocci, Sarcsinia. What are those? What are Vibrio? What are spirochete? What is a glycocalyx? What is flagella? What is an axial filament? What are fimbriae? What are pili or sex pili? What is peptidoglycan? What is an endotoxin? What is selective permeability? What is diffusion? What is facilitated diffusion? What is osmosis? What is active transport? What is tonicity? What is a plasmid? What are inclusions? What are endospores? And lastly, what's the endosymbiotic theory? Now, this is only the major terms. To know all of the objectives of the lesson, you need to take a look at the study guide for this lesson, and it'll have at the very top of it. Oh, it looks like I broke this one out into objectives and then the class notes. So take a look at the objectives for the objectives of chapter 4. Hopefully we're on chapter 4. And then there's the notes for chapter 4. Check to make sure. Let's turn on the slides for chapter 3. All right. Any question about any of that? All right. We'll also be discussing how gram-positive and gram-negative cell walls differ. And then lastly, 3, describe the importance of taxis in bacteria. Prokaryotes compared to eukaryotes differ in that the Greek word for prokaryotes means actually pre-nucleus. Prokaryote, pro means pre, and then karyote means kernel, and they're taking that to mean nucleus. Eukaryote, eu means true, and so it's Greek for meaning true nucleus. The prokaryotes generally have one circular chromosome, meaning in a circle, the beginning spot is the ending spot. In a eukaryote, we normally have a linear chromosome, like the X chromosome. It begins here and ends here. The beginning and ending spot differ. The prokaryotes have their chromosomes not in a nucleus, and the eukaryotes do have their chromosomes in a nucleus. You could also say there's only one generally circular chromosome in prokaryotes, and in eukaryotes, there are generally several to many linear chromosomes. The DNA of prokaryotes, or I should say bacteria, is not associated with the histone protein. The DNA of eukaryotes is associated with the histone protein. In our DNA, we have a big protein, and the DNA actually wraps around the histone protein twice. So histones are the major proteins in the chromosome of a eukaryote, and that protein is totally absent in prokaryote cells, or at least bacteria. Uh, Prokaryotes have no organelles. By organelle, I mean membrane-bound organelle. And eukaryotes do have membrane-bound organelles, like the mitochondria, the chloroplast, the Golgi body. Peptidoglycan is found in the bacteria cell wall. It is not found in any cell walls or any cells of eukaryotes. Prokaryotes generally divide by binary fission. We'll talk a little bit more about that later, but binary fission is where the cell literally divides down the two, making two cells. In eukaryotes, they divide by mitosis. Any question about the comparison? So in the chapter summary, we're going to talk about cell shape and growth arrangement. We'll then move on to cell structures and then talk about the movement of material across membranes, which for prokaryotes is the cell membrane. We'll then talk about eukaryotes, talk about cell structures of eukaryotes, and probably I will not discuss much of this lesson. We'll see how much time we have. And then we'll end up talking about the endosymbiotic theory, which talks about how the eukaryotes gain certain organelles. All right, talking about prokaryote cell size and shapes. The average cell of a prokaryote, actually the average is about 1 to 2 micrometers, but you can have them smaller, down to about 0.4 micrometers. They go smaller than that, but there's a question about the smaller ones, think cells smaller than 0.4, 0.4 micrometers, if they're even viable, that they may divide, but after division, that those small cells die. And there's a question about that. I don't know if it's ever been answered. And then they can get up to about the very largest, 10 micrometers in length. In diameter, they're not that large, but there are some very large prokaryote cells. Getting 10 micrometers in diameter would be the size of the eukaryote cell. The basic prokaryote shapes they come in three basic shapes: round cells, which we call coccus, rods, which we call bacilli, or bacillus for singular, and then spirals, an S-type shape cell. Those are the three basic shape of prokaryotes. There are some other shapes. We'll talk about just one now, and that is a vibrio. You can call a vibrio either a vibrio, or you can call it a bent rod, or you can call it a half of a spiral. Either way, uh, for the spirals, there are basically three shapes: the vibrio, if you count that, the spirillum, also called the spirals, and then the spirochete. There's a few differences between the spirillum and the spirochete. Obviously, the spirillum is really only S shape. It's also a rigid structure, more rigid than the spirochete. The spirochete is more flexible and, I don't know, malleable. And then, of course, the spirochete has, I don't know, many S shapes or many bends to it. These are different species actually that form a spiral and a spirochete, but we lump them together as altogether a spiral for the three basic shapes. There are some unusual prokaryote shapes, like the star-shaped Stella bacteria, and this is actually an actual electron microscopic image of the Stella bacteria. There's the artist's rendition. And then the square or the rectangular Haloarcula, which is an archaebacteria, and then the artist's rendition. Unusual shapes. Most bacterial species are monomorphic, meaning they have one shape, like the Stella. Oops, it's telling me my internet connection is unstable, so let me know if I need to repeat something.
We could not hear the entirety of kind of what you just got done saying.
All right, there are unusual shapes of prokaryotes. The stella are actually star-shaped. That's an actual electron microscopic image of the stella. And then there can be some other shapes like the Haloarcula, which is an archaea or archaea bacteria. It's actually rectangular. They say square type, but it's actually rectangular. Most bacterial species are monomorphic, meaning all of them have the same shape, like the Haloarcula, which is actually archaeobacteria, not bacteria. They're all um, rectangular. The Streptococcus species, they're all cocci. So most bacterial species are monomorphic where they have a single shape, but there are a few species which are pleomorphic, which have many shapes. Like this one here, that almost looks like a cocci. That one kind of looks like a rounded rod. That one, I don't know, what do you call it? A rod that's a bit truncated in the center. And that looks like a dumbbell. That also looks like a dumbbell. That one's real unusual. Reminds me of, what is that? Not Gorn, but Tholian, two Tholian ships that are connected together. We won't talk about that. My point is, there are some species that are pleomorphic. Most of the pleomorphic bacteria are bacteria that do not have a cell wall, and therefore the cell takes on many different shapes because it depends on where the cytoplasm and the cell membrane are. And if you know, the cell membrane is flexible. But most of the bacteria do have a cell wall, and that's why they're one shape, monomorphic. Didn't we just talk about that? Oh, I did. I went the wrong way. All right, so that is the bacteria shape. Let me show you one other usual shape. I don't know why it's down here because Coccobacillus is not a cellular growth arrangement. Coccobacillus is a shape. It is in between a bacilli and a cocci. And that is if you were to get a bacillus that's rounded on the edges and then shrink it, you will get a coccobacillus. Or you can, let me blow that up. take a cockeye and elongate it. That one actually looks like a cockeye, but you can elongate it, and that's a Coccobacillus. That is a cell shape. It is considered one of the three shapes because this is a combination between a cockeye and bacilli. It is not a cellular growth arrangement, so it should not even be in this slide, but this is the way the author made this slide. I don't know why they have that there. Like I said, it's not a growth arrangement. Talking about the cellular growth arrangement, with the cockeye, you can have a singlet, a single cockeye, which actually that one is, and so is that one. Let me see if I can find one. That one right there is a singlet. That's kind of a singlet there. You can have cocci, which are two cocci connected together, and that we call a diplococci. And if you extend this diplococci, you'll get a streptococci, and that's cocci in a row. You can have four cocci stuck together, and that's called a tetrad. You can have eight cocci stuck together, and that's called the sarxinia. And these are hard to see because there's so many of them. But there's one, two, three, four cocci in a tetrad. This is an actual electron microscopic image. And then the sarxinia, that's about eight right there. You can't see, well, certainly you can't see that one. You can't see this one either because it's behind the others, but there's eight cocci there. And there's almost eight. There's seven there, and then there'll be one shown behind these. So that would be the eighth. You can take a look at the plane of cell division. This one divides only in one plane, why it forms diplococci or streptococci, and this is just when this one splits here into two, that one then splits into two, and then that one splits into two, making a streptococci. And then the tetrads have two planes of division along the y-axis and the x-axis. The sarxinia and the staphylococci have three planes of cellular division. Let me show you the staphylococci is a cluster. Staphylococci actually means cocci growing in a cluster like grapes. That's what staphylo means, cluster like grapes. And if you put a stem on there, that looks like a cluster of grapes. Those are the different growth arrangements that a cocci can have. Bacilli have fewer growth arrangements. Let's put that over there. You can have a singlet, a single bacillus, and that would be one there, and that would be one there. There's one there. You can have a diplobacilli, and that's two bacilli stuck together, which for the life of me, there's a diplobacilli right here. I don't see any in there, but right here, that's a diplobacilli. And that could be a diplobacilli right there, with this being the division right there. You can have streptobacilli, and these are bacilli that are hooked together on the long end, sort of looking like a sausage link. And there's a streptobacilli there. Only three of them, but that's good enough. That's four, that's better. And then the other bacilli hooked together in not called the streptobacilli link, but where they're hooked together on their long side, we call that a palisade, where one bacilli is this way connected to several on the long side. Let me stop this. Those of you who are familiar with Mark Twain, not Mark Twain, Huckleberry Finn, Tom Sawyer and Huckleberry Finn, those two books, you'll know that Tom Sawyer was painting a fence in the book, and he tried to convince Huckleberry Finn that it was fun painting it so that Huckleberry Finn would do the painting for him. And there are fences with I can't draw that very well, but you get the idea. There are fences with a board stuck lengthways this way, and then I guess lengthways that way, but the long side that way. I'm not drawing this very well, but you're getting the picture. And then the bottom would be the ground. And then you just stick the boards in the ground. And then you have your palisade fence. It used to be very common, especially in the past. They're not so common anymore. I can occasionally find them more in cities than farmers using them. Anyways, that's a palisade, named after the palisade fence. Any question about the growth arrangement of bacilli and cocci? All right, before I go on, let me state that the growth arrangement is always pleomorphic unless the cell only has singlets. If the species only produces singlets, then that's all the species will have. But if it's any other growth arrangement, it will have multiple growth arrangements. Like Diplobacilli, the two can break apart, and so you'll see some singlet bacilli and some diplobacilli. The strain was same with streptobacilli. If this were to break right there, you then have streptobacilli and diplobacilli, and if that breaks, you'd also have singlets. For the staphylococci, one could break off, and then you have a singlet. Two could break off, and then you could have a singlet and a diplobacilli and a staphylococci. A cluster could break off in a Sarxenia, and a smaller cluster can break off and it's a tetrad. Occasionally you can see it'll break off really weirdly, and you can see a streptococci among the staphylococci. And then you can also have clustering where the cells start clumping together, where you have a whole bunch of streptococci clumped together, and then it'll look like a staphylococci. So the growth arrangement is pleomorphic with one exception, and that is if the species can only produce a singlet, then you'll only see singlets. There actually are some species that only produce singlets. Most of them are the spirochetes. The spirochetes are always nothing in cellular growth arrangements other than a singlet. Any question about the cellular growth arrangement? Don't get it confused with the cell shape. All right, this is the structure of a typical bacteria cell, prokaryote cell. All cells have three parts. That is, they all have a plasma membrane, also called the cell membrane. They all have a cytoplasm inside the cell membrane. And then they all have DNA as their genetic molecule, their genome. So all cells have those three parts. All cells also have ribosomes, but please understand that the prokaryote ribosome differs from the eukaryote ribosome. The eukaryote ribosome is larger than the prokaryote ribosome. So these structures marked in red are structures that you can find in all cells. All the other structures you can only find in certain cells. Like the inclusion is only found in certain prokaryote cells. To my knowledge, there's never an inclusion in a eukaryote. You can find the capsule. It's something secreted by the cell. It's exterior to the cell. If you can see it under the microscope, it's called a capsule. If you cannot see it under the microscope, then it's called a slime layer. There's a little difference in the two, and we'll talk about those later. All right, you can see cell wall in many cells. Most bacteria, most archaebacteria do contain a cell wall, but not all of them. Plant cells and fungi all have cell walls. Protists, most of them have a cell wall, but not all of them. The animal-like protists lack a cell wall. And then animal cells do not have a cell wall. Any question about that? The DNA in prokaryotes differ from the DNA in eukaryotes, which I don't have that shown. The DNA in prokaryotes is wherever it floats in the cytoplasm. There is no distinction between where the DNA is and where the cytoplasm is. There's no boundary between the two. So wherever the DNA floats, and the DNA is generally in suspension in the cytoplasm. So wherever it is, that's where the DNA is. And the cytoplasm is the same wherever the cytoplasm is. The cytoplasm is everything in a prokaryote underneath the cell wall or the cell membrane. You can find plasmids in bacteria and archaea and yeast too of eukaryotes, but especially in bacteria you can find it. A plasmid is a DNA molecule outside of the chromosome. The plasmid is also much smaller in size than the chromosome. You can have flagella for some cells. In bacteria, if the cell can move or swim, it has at least one flagellum. Fimbriae are the hair-like structures coming out of the cell. Cells may have fimbriae. All cells have a plasma membrane. I already talked about that someplace right there. And the cell wall is exterior to the plasma membrane. Most bacteria have a cell wall, but not all of them. I already talked about a capsule. All right, any questions about the different structures of a prokaryote cell? Let's talk a little bit about the glycocalyx. The glycocalyx is secreted by many prokaryotes. Obviously, not all, not even the majority, but many do secrete a glycocalyx. It is outside the cell wall, so we do not consider it a part of the cell. It's usually sticky. You can find the glycocalyx in three different types. A capsule, which is a neatly organized glycocalyx. It has molecules in it, so it's more than just water. The capsule can be seen by a light microscope, and the capsule can help prevent phagocytosis of the bacteria from a white blood cell. So if a bacteria produces a capsule, the white blood cells cannot, at least easily, phagocytize them. Usually the bacteria with the cell capsule escapes phagocytosis and then just can grow, and if it needs to move and all that. Anyways, that's a capsule. Remember, it's neatly organized. It has more molecules in it than just water. You can see it with the light microscope. The slime layer is an unorganized glycocalyx. It's mostly water. It's a loose structure held loosely to the cell. It too is secreted by prokaryotes outside the cell wall. You cannot see the slime layer with a microscope. To see the slime layer, you have to use an electron microscope. But the slime layer and the capsule are easily removed by normal staining procedures. So if you're performing a normal stain, like the Gram stain, you're likely to wash away the glycocalyx, and then you won't be able to see it. There's a third type of a glycocalyx, and that is only found in biofilms. This glycocalyx is an extracellular polysaccharide, which is secreted by the cells of the biofilm. And it's actually this extracellular polysaccharide which makes the biofilm. The biofilm is something secreted by the cells that encases the cell within the biofilm. And cells that are in biofilms are usually much harder to kill than cells that are outside the biofilm, meaning an antibiotic can be very readily kill all the cells of a certain species that does not grow in a biofilm. But once it starts growing in a biofilm, that same antibiotic may not be able to remove all of the bacteria that cause the initial infection. All right, any question about the glycocalyxes? The flagella is the structure seen in bacteria, as well as archaebacteria, that allow the cell to move or to swim in a liquid media. So the flagella is used for locomotion, and in prokaryotes, it's the only way that cells can move or swim. Eukaryotes have additional ways to move. They can move if they have flagella on them. The human sperm is one example of a cell that has a flagellum, and obviously it can move. There are some other eukaryote cells that have flagella. Oh, this is what I was trying to say. That's one way that eukaryote cells can move, by the flagellum. The second way they can move is by cilia. And then the third way they can move is by pseudopods. Usually, a eukaryote cell will pick one way to move, and then for the rest of its life, that is the way it will move. Like a white blood cell can move by phagocytosis, and that's the only way, as far as I know, a white blood cell can move, other than passive movement, like if the white blood cell were to be moved around by the blood. But by its own movement, it has to move by pseudopods. All right, any question about flagella? Flagella is mostly made up of the protein flagellin. Flagella are mostly outside the cell wall. So there's the flagella there, flagellum here. And it's mostly outside the cell wall, but there is a small portion of the flagella which is inside the cell wall. And there's even a small portion that is inside the cell membrane. But the vast majority of the flagella is outside the cell. Any question about any of that? All right, you need to know the four basic arrangements that bacterial flagella can have for the lab. You can have monotrichous, shown here, amphitrichous, which you have one or several on one side, I guess one on amphitrichous, lophotrichous, several on both sides, and then peritrichous, uh, many of them flagella all over the cell. I will not quiz you on the flagella arrangement, but you do need to know it for answering one of the questions on the quiz. Not the quiz, on the lab worksheet. Any question about flagella? All right. Motility in cells happens because of flagella allowing the cell to run or tumble or to swim. And by tumbling, what we say is that the cell, let me make it a rod right here, can then tumble. So then it'll be up like this, and then can go down, fall down, and then down here. And that is taxis, where the cell can move. Taxis just means the movement either toward or away from a stimuli. In bacteria, we have two types of taxis, chemotaxis, and the second being phototaxis. Chemotaxis means the cell responds to some chemical. Doesn't matter if it swims towards the chemical or away from the chemical. Either way, it's chemotaxis. The cell moves in response to a stimulus, which in this case is a chemical stimulus. You can also have phototaxis, where the cell either moves towards or away from the light. And this one, the light source is up here. The cell is moving this way, so it's moving away from the light, and that would be phototaxis. To move, the cell needs flagellum under phototaxis, as well as light. Any question about any of that? Axial filaments, also called endoflagella, are flagella which are inside the cell wall. Inside the cell wall of a cell, we find axial filaments or endoflagella in the spirochete. So here's the spirochete, and there you can see the endoflagella wrapping around the cell and going all around the cell. And right here, we have the same spirochete showing the axial filament or the endoflagella wrapping around the cell. It's shown in the darker pink, meaning the purple is the spirochete, the pink is the axial filament, and it just wraps around the cell. But this wrapping is actually inside the cell wall. Let me get that out of here. Blow this up a little. Oops, wrong one. So the light pink is the cell wall, running around the cell and then running around the axial filament. The dark pink is the axial filaments themselves. And you can see they're definitely inside the cell wall, which is the light pink. Oh, this has separated the cell wall by the outer sheath and the outer sheath of the cell wall and the peptidoglycan layer, which is the dark purple. It is between it is between the outer membrane of the cell wall and the peptidoglycan. That is where the endoflagella is made. I won't quiz you on that, but some people try to state that it is outside the cell wall. And no, the outer membrane of the cell wall is a part of the cell wall. So it's inside the cell wall. Spirochete have a unique motion where they actually swim in a spirochete type motion. Let me see if I got that up now. Oh, there it is. Just real slow. I don't know why the network is so slow now. Looks
As I'm
like
in
we
the
have a
midst of
commercial.
applying for residency right now, wish me luck. Grammarly has again been a lifesaver with my application in helping me write.
So just to change direction, it just keeps, it moves in the different direction by simply spinning in the opposite direction. Spirochete have a curious corkscrew-like movement. Let me start all over again. Moving like a corkscrew. And spirochetes are very flexible. There you can see the spirochete. What it did was it bent, I don't know, together the two ends, and then changed its direction from moving north-south to east-west. Any question about the spirochete? There you're seeing the corkscrew-like movement. More corkscrew-like movement. This is going down. Let me shrink that down. Move down a little further. Fimbriae, which you can't see there, I got it now, are shorter and straighter and thinner than flagella. Fimbriae are used for attachment to a surface. Fimbriae are not used for swimming. But here the yellow is the fimbriae, and they tend to hold on to something on the tips of their long, I don't know what you want to call those, not rods, but appendages, I guess. So it attaches to things using the fimbriae. The fimbriae do not serve in swimming. The pili are similar to the fimbriae with one exception, and that is well, two exceptions really. The sex pili are very much larger and longer than the fimbriae. So here we see a cell. Let me blow this up for you. And these right here are all fimbriae. That, on the other hand, is not a fimbriae. That is a sex pilus, or pili if there's more than one. Oh, let me get that over there. There we go. So they are longer than a fimbriae. They're also hollow. Sex pili are used in transferring DNA from one cell into another cell. We'll talk more about that later. Any question about?
We can't see your screen. I think it froze.
Oh, really?
Oh, there it goes.
I don't know what that is. It must be my internet connection. Because I didn't change it. It just did that. All right, let's go on. So the sex pili are hollow. They allow a plasmid to leave this cell and enter that cell, and they are longer than fimbriae. The prokaryote cell wall is complex, semi-rigid structure. It is responsible for giving the cell its shape if the cell has a cell wall. The few bacteria that do not have a cell wall take on many different shapes because the cell wall holds the cytoplasm together so the cell doesn't burst. And if there wasn't a cell membrane, water would just continuously move into the cell. The cell would continuously expand until it could expand no further. And then the water would move into the cell, causing the cell to burst. And then the cell inside ingredient will spill out and the cell will die. So I mentioned the cell wall can help protect the cell from osmotic lysis. It can help protect the cell in other ways too, but like preventing antibodies from binding to the antibody. The prokaryote cell wall is located outside of the cell membrane. And even though the cell membrane is considered the gatekeeper, allowing certain molecules to come in and then allowing other molecules to leave, and any molecule it either does not want to enter the cell or a molecule it doesn't want to leave the cell, we have the plasma membrane, which can control that. My point being is that the cell membrane should be considered the division between the inside and outside the cell, but it's not. The cell wall is actually the dividing part of the cell in between the cell membrane and the outside of the cell. The cell wall can help prevent the cell from getting osmotic lysis. During osmotic lysis, water will continue to move into the cell. If the cell lacks a cell wall, the cell membrane will expand. Water will continue moving into the cell until the cell membrane eventually will burst. It's like 40 to 45 seconds when you put red blood cells in distilled water. That's because they just can't handle the tonicity. Cell wall is located outside the cell membrane, but remember it is considered a part of the cell. So the cell wall is considered a part of the cell. Anything exterior to the cell wall is considered outside the cell. The cell wall can contribute to the pathogenicity of some species, like the Mycobacterium species. Their cell wall can contribute to its pathogenicity. Most peptidoglycan is made from bacteria. So most peptidoglycan that we, I guess, would buy in the store or something, it's coming from being made in bacteria. Peptidoglycan is a polymer of a repeating disaccharide. The two sugars which repeat to make the peptidoglycan are NAG and NAM. NAG stands for N-acetylglucosamine, and NAM for N-acetylmuramic acid. You can know NAG and NAM by N-acetylglucosamine or by NAG, it doesn't matter. I don't care which one you know, but you should know one or the other. The point being is that NAG and NAM are a disaccharide and they're linked by a covalent bond holding them together, as well as a peptide bond. We'll talk about the peptide bond in just a minute. So there's NAG and NAM, a better picture than here, showing you that it's just one sugar. Start with that one because you can read it better. After the other, one sugar after another, one sugar after another, making a whole row of NAG and NAM in the cell wall. In the cell wall, we have multiple rows next to each other. And then we have multiple rows underneath this. And if this is not the outer one, there are multiple rows on top of it too. And what's holding the peptidoglycan together in the row, it's a chemical covalent bond in between the two molecules, probably a carbon and hydrogen, but it could be a carbon and oxygen. I'm not sure what the linkage is between NAG and NAM, but my point being is they're linked together in a chemical bond, making several rows. The rows are not connected together by anything other than the peptide bond. And that is, let me see if I can show it right there. This row and that row are held together by this short peptide. And it is true that this row and that row are held together by that peptide, holding that NAM to either that NAG or that NAM. I'm not sure where it goes. And how this short peptide holding this row to that row holds together is this short peptide is linking this peptide and this peptide. Let me warn you that this artist rendition is showing you one layer of peptidoglycan. In reality, there's multiple layers. This one's a little better. It's showing you 3 layers. But of the gram-positive cells, they will have a peptidoglycan layer that is at least 2 layers thick. So it's more than 1 layer. It's something like 3 to 20 for a gram-negative cell. That's how many layers a peptidoglycan is. And then for a gram-positive bacteria, they'll have more than 20, up to at least several hundred, maybe several thousand layers of peptidoglycan. Penicillin actually inhibits the lattice formation, meaning normally when we're putting down cell wall, we'll make the NAG and the NAM, and then we will link it, this NAG and NAM row to that NAG and NAM row with this peptidoglycan, and then the same with this row of peptidoglycan. It'll be linked to this row of peptidoglycan by a short polypeptide chain. And then you can link this peptide to that peptide by putting in another short peptide chain, and that will link that chain to this chain. Penicillin inhibits the lattice formation. It will prevent the polypeptide from forming right here in between this polypeptide and that polypeptide, holding this NAG and NAM chain to that one. So in the case of penicillin G or penicillin V, it'll intercalate into the cell wall and then put itself right here. And then this short peptide can't link. And that will always make a kink in this molecule of DNA, where this one will have at least one region right here, where it's not connected to this region of NAG and NAM, and that will put a hole in the peptidoglycan. The hole is important because the cell membrane can protrude out of the hole, and then as more water moves into the cell, the cell membrane will continue to expand out of that hole until the cell eventually will pop, and then that'll kill the cell as the cytoplasm is released. And this hole in the cell wall is very much like blowing up a balloon inside of a box with a hole in it. The balloon will begin protruding out of the hole. And then as you continue blowing up the balloon, that section of the bacteria with this cell membrane, I'm trying to remember the name for that, and it'll just continue to protrude out of the hole. More water will move into the cell. The cell membrane will continue to extrude out of that hole in the box, much as if you're blowing up a balloon, until eventually the cell membrane can't expand anymore. And then that'll, when more water moves into the cell, that will destroy the cell membrane, as well as release the contents of the cell into the environment, and then the cell will die. So penicillin G stops the proteins, the short peptides, from linking together in the cell wall. And this also explains why penicillin G may not work on a gram-positive cell, and that is penicillin G can only work when the cell is putting down its peptidoglycan. If the cell already has its peptidoglycan down and then receives treatment, which is penicillin G, the penicillin G cannot intercalate in between the rows of peptidoglycan and then block this peptide from forming because that peptide is already formed, linking this row of nag and nam together to that row of nag and nam. So penicillin has to work on cells that are actively dividing. If the cell is not dividing, penicillin G will not work on it. Any question about any of that? All right, let's look at the cell wall in a little more depth. Here we're seeing a gram-positive cell, and there's the cell membrane, that yellow. The purple, very thick, is the layers of peptidoglycan, shown greater here, but let me take a look here. You can kind of see the layers of peptidoglycan in there. Maybe I'm misinterpreting it, but I kind of see it. And it's layer upon layer. The gram-positive cell wall will have something like more than 20 layers of peptidoglycan, up to several hundred, maybe even several thousand layers. This artist rendition is showing you the cell wall, but not in reality. It's only showing you 3 layers of peptidoglycan. You find more than 3 layers in a gram-positive cell. It's also showing you there are other molecules besides peptidoglycan in the cell wall of bacteria. Peptidoglycan is the main molecule, so it is the majority of the molecules in the cell wall. But we can also have teichoic acid, which kind of helps stabilize and link the peptidoglycan layers together. You can have lipoteichoic acid, and this will be the same teichoic acid in the cell wall, but the lipoteichoic acid can also go through a region of the cell wall which is fat soluble, meaning a fat that had fat that you could refrigerate. I don't know how else to word that. In it, the lipoteichoic acid can be in the cell wall where the ionic and the ionic and the other interactions between the amino acids of the peptide will be, meaning where they're interacting with each other, and that would be, let me go back here, where this one is binding to that peptide, which is binding to that peptide. The lipoteichoic acid can be in the cell wall where there are lipid regions in the cell wall, as well as in the cell membrane, which is mostly lipid. And it's the lipid portion of the lipoteichoic acid which is in the cell membrane or in the cell wall where there's a lipid region in the cell wall. Most of the cell wall is peptidoglycan, in which case the teichoic acid part would be in the peptidoglycan part. So remember, in the gram-positive cell, we have a very thick layer of peptidoglycan, with peptidoglycan being the main molecule in that layer, and it's several layers between 20 and several hundred or maybe several thousand. Let's take a look at the gram-negative cell. Nope, gram-positive. So I'm just reiterating, we do find lipoteichoic acid and teichoic acid in the peptidoglycan. The teichoic acid may help regulate the movement of cations through the cell wall. It's not required because a cation is dissolvable in water, and anything that is dissolvable in water will come right through the peptidoglycan of a gram-positive cell. So in the gram-positive cell, an ion or anything can move right through the cell wall because it can dissolve in water. But the teichoic acid, the ions, the cations, may move down the teichoic acid, helping them move into or out of the gram-positive cell wall. The cell wall can also have some polysaccharides, which can provide antigenic variation among the cells. The gram-negative bacterial cell wall differs from the gram-positive cell wall. Here we're looking at the gram-negative cell wall. It does have this yellow layer below it, which is not part of the cell wall. It's actually part of the cell membrane. And then it has a thin peptidoglycan layer shown in purple. And then it has a rich lipid layer outside the peptidoglycan. This lipid-rich region outside the peptidoglycan is called the outer membrane of the cell wall. So it is part of the cell wall, along with the layer of peptidoglycan. The outer membrane is important. You only find it in gram-negative cells. This layer of the outer membrane, meaning the bottom layer of the outer membrane, is actually fundamentally similar, if not the same, as the layers of phospholipid in the cell membrane, meaning the majority of the molecules in the cell membrane are phospholipid. That's also seen in the outer membrane, where the majority of the molecules are in the outer membrane are phospholipid. Any question about any of that? We do have other molecules, particularly other lipids, in this membrane, meaning the outer membrane of the outer membrane. So the inner membrane is mostly phospholipid. The outer membrane is also mostly phospholipid, but it does have other lipids. That big blue blob there is actually lipid A, and it is a lipid. It's also a endotoxin. Exposure to toxins is a bad thing because something that is a toxin is toxic to us. So all gram-negative bacteria that cause an infection where the species has a cell wall will always contain lipid A, an endotoxin. And the patient's just going to have to deal with being exposed to a toxin because there's no way to not expose the patient to it if he has a gram-negative infection. The lipid A is actually linked to an O polysaccharide, making lipid A and the O polysaccharide a lipopolysaccharide. Also in the outer membrane, we have molecules, porin protein. The porin protein acts as a channel to allow molecules the cell wants in the cell to come through the porin protein channel, go right through the peptidoglycan layer of the cell wall because it's water soluble, and then come to the cell membrane. If the molecule does not readily diffuse through the cell membrane, then it has to get through the cell membrane by an active transport protein. And the cell will make one active transport protein to move one molecule into the cell. So the first thing, let's talk about my red blood cell, and it doesn't have, I don't know, what am I trying to say here? So we have a bacterial infection in me, it's gram-negative. it will have to, I would have to deal with the endotoxin because there's no way to kill this bacteria without releasing endotoxin. And obviously, you want the patient to get rid of the infection, so you give antibiotics to get rid of the gram-negative bacteria, and that will release lipid A, a toxin, into the patient. But it's better to expose the patient early to the lipid A than later, because later there might be more bacteria, and the more bacteria there are, as long as the bacteria have a cell wall, the more lipid A the patient would be exposed to. The porin molecule, like I said, allows many molecules that the cell wants to enter the cell through the outer membrane, as well as it allows many molecules to exit the cell to get through the lipid bilayer of the outer membrane. That molecule will move through the porin protein. So the porin protein acts differently than the proteins in the cell membrane, because in the cell membrane, we have one active transport for every molecule that the cell wants to move across the membrane. Meaning if we have glucose outside the cell, we will make an active transport protein to move the glucose into the cell. If we have lactose outside the cell, we'll make a separate active transport protein to make the lactose move inside the cell membrane. But both the sugar lactose and the sugar, what did I say, sucrose, can move through the porin protein to move through the outer membrane. Because neither sugar, they're both water soluble, would get through the lipid bilayer of the outer membrane. And you need to have a transport protein to move it through the cell membrane, because the molecule cannot move through the lipid bilayer of the cell membrane. Any question about any of that? This is just showing it in greater detail. You can have other proteins in the cell wall. Like there's a lipoprotein. You can have a lipoprotein in the cell wall as well. And the lipid portion of this protein will be either in the outer membrane of the cell wall or in the cell membrane. So in the gram-negative outer membrane, we find different molecules, phospholipids, the main one. We also find lipoproteins, lipopolysaccharides. You don't need to know this. I won't quote you on you there, but the space between the outer membrane and the plasma membrane is called the periplasm, and that is where we find the layers of peptidoglycan. The gram-negative outer membrane can offer some protection from host defenses, like the outer membrane can prevent the complement protein from binding to the cell, which is causing the infection. The outer membrane can also help prevent antibiotics from binding to the cell membrane or the cell wall. The outer membrane also has O polysaccharide antigen, which gives the different species an antigenic differentiation. Like I'm sure most of you have heard of E. coli O157H7. This antigen, it's actually two, the O157 is one, the H7 is another antigen. This E. coli is a human pathogen. Most people who get it will get gastroenteritis, and some of them will get diarrhea. Normal E. coli lacks these two antigens. And normal E. coli is not a human pathogen. At least in your gut, it belongs there. Let me remind you, lipid A is an endotoxin. Exposure to a toxin like endotoxin is always bad for a patient because a toxin means toxic, and so all endotoxins are toxic to us. Endotoxin comes from the outer membrane of the cell wall of a gram-negative cell. So you only find endotoxin in a gram-negative cell bacteria that has a cell wall. We already talked about porin proteins acting as channels to get molecules into and across the cell membrane. All right, any question about any of that? I guess I'll go a little further. Here's a comparison of the gram-positive cell wall. Thick peptidoglycan, a rigid structure. The gram-negative cell wall, a thin peptidoglycan, not as rigid. It is rigid, but not as rigid as gram-positive. We find teichoic acid in gram-positive cells. There's no teichoic acid in a gram-negative cell wall. There's no outer membrane in the gram-positive cell wall. There is an outer membrane in the gram-negative cell wall containing lipid A and endotoxin. Any question about any of these? I guess I could put in here, you can find a porin molecule in the outer membrane of a gram-negative cell. There's no porin proteins in the cell wall of a gram-positive cell. The cell wall does make the cell stain differently when applying the gram stain. The crystal violet iodine complex forms crystals which will form a complex in the cell wall of a gram-positive cell, meaning the crystal violet iodine crystals complex do not leave the thick peptidoglycan layer of a gram-positive cell, but they do leave the thinner peptidoglycan layer of the gram-negative cell. Alcohol of the gram stain can also dissolve the outer membrane, which is mildly important because if the outer membrane is intact, the molecule trying to leave the cell will have trouble leaving the cell. But usually that molecule can go out the porin protein of the outer membrane. And then lastly, the gram-negative cells stain pink because we apply the dye safranin to color the clear or decolorized gram-negative cells resulting from the gram stain. Any question about any of that? All right, this is a summary table of gram-positive and gram-negative cells. You only need to know the portion above the red. You can ignore all the portion below the red. The gram reaction of a gram-positive cell is positive, gram-positive, because it retains the crystal violet iodine complex of the gram stain. The gram-negative cells are gram-negative because they decolorize when you apply the decolorizing step. It'll pull all the color out of the cells. And then they turn pink after being stained with the dye safranin. Peptidoglycan layer is thick in the gram-positive, thin in the gram-negative. Note this is a mistake. The author said it's a single layer. It's actually like 3 to 20 layers of peptidoglycan. Teichoic acid present in gram-positive cells, absent in gram-negative cells. The periplasmic space absent in gram-positive cells, present in gram-negative. The outer membrane absent in gram-positive cells, present in gram-negative cells. Lipopolysaccharide of the gram-positive cell, it's virtually none in the gram-positive cell. You have a high content of lipopolysaccharide in the gram-negative cells. All right, any question about any of that? If not, I think I'm going to stop right here and we'll pick up from this, oops, slide next time. I don't know where I'm going to put that in here. There we go. All right, if there's no questions, I'm going to log out and I'll see some of you in the lab at 6:30. How do I get rid of this?